The overall goal of this procedure is to evaluate the stem cell properties of human ovarian carcinoma cells using multi- and single-cell-based spheres assays. This is accomplished by first selecting SOX2 reporter RFP lentivirus transduced cells by puromycin treatment. In the second step, the RFP positive and RFP negative cells are isolated and plated by FACS as single or multi-cell colonies per well. Next, the primary sphere formation is analyzed microscopically, and then the primary sphere cultures are dissociated and replated for analysis of the secondary sphere formation. Ultimately, these sphere assays are used to calculate the proportion of putative ovarian cancer stem cells that display sphere-forming capacity.
This method can help answer key questions in the cancer stem cell field, such as what are some potential cancer stem cell populations and what is their frequency within the whole tumor bag.
Although this method can provide insight into ovarian cancer stem cell identification, it may also be applied for isolating cancer stem cells from other solid tumor entities such as breast carcinoma.
"24 hours after transducing human ovarian carcinoma cells with lentiviral particles, remove the viral supernatant and wash the cells in PBS. Culture the cells in complete medium for 48 hours. Then change the medium and add 10 micrograms